It is the small difference in base pair sequence of DNA that makes the phenotypical appearance of each individual unique. Human genome have about 3 million base pairs. Therefore, it could be a time-consuming and difficult task to find out genetic difference between two individuals, as it involves comparison of 3 billion base pair genome. Easier and quicker way to compare is DNA fingerprinting, which is the test to classify and analyze DNA or genetic information. In human being, 99% of DNA sequence are identical and are known as bulk genomic DNA. Remaining 1% of DNA sequence varies and are present as a stretch of a repetitive DNA sequence. DNA fingerprinting identifies the difference in this region. Blot is used to verify the presence or absence of a specific nucleotide sequence in DNA from different sources, and to identify the size of the restriction fragments that contains the sequence. In this process, the DNA is isolated from each source and digested by specific restriction enzyme. DNA restricted fragments are then loaded onto the agarose gel, the fragments separated by electrophoresis according to size, with smaller fragments migrating faster than the larger fragments. Fragments are made visible using ETBR. DNA is then transferred from a fragile gel to the nylon membrane. Then the radioactive labeled probe is added. The probe binds to the complementary DNA segment. Note that the DNA segment being probed is not present in organism B. To detect the position of the radioactive probe, the nylon membrane is covered with the X-ray film. The position of the probe becomes visible. Similarly, DNA fingerprinting is done in plants. The only difference is that the sample collection. For easy convenience, leaf samples are used. The other procedures all remain the same. Now, see here. When a child is born, they inherit 23 chromosomes from mother and 23 chromosomes from the father. Child 1 and child 3 are the children of both mom and dad. You could see the colors here. Child 2 is a child of mom but not a child of dad. Child 4 is not a child of mom or dad. Identical twins have same DNA but have a unique fingerprints. Isn't that strange? DNA fingerprinting is not at all a sequencing technique. Rather, it identifies repetitive sequence present in one's genome. Such repeat sequence also vary in length due to variation in the number of repeats, hence called variable number tandem repeats. Now, this first one is the session of victim DNA. There are 3 regions of repetitive DNA. Session of suspect DNA. The same 3 regions of repetitive DNA are present here, but some include different number of repeats. Now let's compare this to the sample. This is the session of DNA from the crime scene here. The length of the repeated sequence matches the length of the suspect's DNA. So the DNA found at the crime scene belongs to the suspect. DNA fingerprinting also helps in phylogenetic analysis. A phylogenetic tree is a diagram that represents the evolutionary relationship among the organisms. Phylogenetic tree are hypotheses, not definite facts. The pattern of branching in a phylogenetic tree reflects how species or other group evolved from a series of common ancestors. In tree, 2 species are more related if they have the more common ancestor and less related if they have the less recent. I hope this video was useful to you. Thank you.